Cadmium is a heavy metal that is closely related to smelting, battery, and metal coating industries. Cadmium occurs as a byproduct of copper refining, since it is often found within copper ore. Cadmium is also commonly used as a metal coating because it does not easily corrode. Production of nickel-cadmium batteries is a significant source of cadmium waste. Cadmium can contain water through direct contact with coated metal, batteries, and sludge from smelting factories. The EPA has set the maximum contaminant level for cadmium at 0.00 milligrams per liter. Contact with cadmium can affect the cardiovascular, respiratory, and gastrointestinal systems. Some effects of cadmium poisoning include cancer, blood pressure spikes, breathing problems, digestive anomalies, and blood clots. Side effects are more pronounced and serious in children since they are still developing. Before starting the lab portions of our project, we trained our new members in proper sterile technique and safety procedures. This is particularly important for two reasons. The first is that our new members have no experience with standard procedures used in synthetic biology, including culturing bacteria and plasmid isolations. Second, our lab space overlaps active classroom space, so careful attention to safety is critical. Because we work in a high school, the materials and chemicals we can work with are restricted to what is considered safe for a school environment. To build the cadmium detector, we used a cadmium responsive promoter contributed by the 2009 Newcastle IGEM team, and the GFP coding region contributed by the 2005 MIT IGEM team. The cadmium promoter is actually a series of two promoters that are each sensitive to several metals. The only sensitivity in common is cadmium. Thus, the GFP is only responsive to cadmium. For our arsenic detector, we used an arsenic responsive promoter contributed by the 2006 Edinburgh IGEM team and the same GFP coding region. The arsenic promoter is not solely responsive to arsenic. It is also responsive to silver, copper, and cadmium. Finally, to build the lead detector, we used a lead-responsive promoter contributed by the 2007 Brown IGEM team, and again, the same GFP coding region. The promoter was originally designed to require a lead-binding protein, which is not available in the parts registry. However, we decided to pair the promoter with the GFP recorder anyway. To construct our biobricks, we digested the cadmium, arsenic, and lead-sensitive promoters with EcoR1 and SPE1, and the GFP recorder plasmid with EXPA1 and PST1. All of the initial plasmids were ampicillin resistant. We ligated each promoter with its own GFP recorder and chloramphenicol resistant PSB1C3 plasmid backbone. We used competent cells and a biobrick assembly kit donated by New England Biolabs in order to create our sensors. One benefit of using the required chloramphenicol vector is that bacteria transformed with uncut original plasmid will not be able to grow. We chose green fluorescent protein as a recorder since it is easy to detect with our UV light box, and we can quantitate it with our new spectrophotometer.
The initial testing protocol for all of our detectors was essentially the same. We grew the bacteria in LV along with varying concentrations of cadmium nitrate or lead acetate. To test our arsenic and cadmium detectors, we used cadmium nitrate with varying concentrations from 0 to 20 millimolar. To test our lead detector, we used varying concentrations between 0 and 20 millimolar lead acetate. However, as we began our tests, we immediately ran into a problem. All of our dilutions, including the negative control, were glowing with roughly the same intensity as seen above. After crunching some numbers and online research, we found that LB, our growth medium, fluoresces naturally. In order to solve this problem, we attempted to use M9, a minimal growth medium. Unfortunately, our cells were unable to grow in the minimal medium. After this, we were still unsure as to whether our detectors worked properly. And after even more research, we found an inexpensive solution called phosphate buffered saline, or simply PBS. We then reverted back to our original protocol of growing the cells, and growing the bacteria in LB, but before measuring fluorescence, we spun them down and then resuspended in PBS. We could then proceed with our quantitative analysis. And in order to account for differences in growth density, we report all of our units in fluorescence units per OD. As shown by the picture above, our sensors were successful. A 1 millimolar concentration of cadmium nitrate is shown on the left, and a negative control on the right. Here we have our results in fluorescence units per OD for our lead detector. While we did see an increase over background fluorescence, it was inconsistent across all of our dilutions. And there are several potential problems here. First, the promoter is reported to require a lead binding protein, which we did not incorporate into this construct. Furthermore, we noticed a precipitate starting at the 0.5 millimolar concentration, and we feel that the lead's toxicity or this precipitate interfered with our results. For our arsenic detector, we had fantastic results. Because we are in a high school, we are not allowed to have or handle any form of arsenic on campus. Hence, we used cadmium nitrate in order to test, since it is responsive to multiple heavy metals. This graph shows that we are able to detect the promoter activity at levels below 1 millimolar. The next step of our testing procedure will be to further quantitate the minimum detectable level. And lastly, we have our cadmium detector. We used cadmium nitrate in the same testing technique mentioned previously. Our detection level is somewhere between 1 and 5 millimolar. Even though there is a slight increase at 1 millimolar, the results were statistically insignificant. As with the arsenic promoter, further testing will determine the actual minimum level of detection. And the table above shows our sensitivity levels in comparison with the federal limits. While the federal limits are below 0.01 parts per million, we are currently only able to detect down to, at best, 240 parts per million. This means that you could probably detect a problem long before our sensors could. And clearly, for these detectors to be practical, we will need to increase the sensitivity by several orders of magnitude.
At last year's competition, we attended the UC Davis team's presentation in which they described a protocol from the genetic PCR. Small changes in promoter sequences can result in altered promoter sensitivity. We plan to use this process to increase the sensitivity of our detectors. An alternative to this procedure would use tuners created by the 2007 Cambridge IGEM team. In the spring, we plan to incorporate these parts into our fireworks. Along with the increase in sensitivity, we will further investigate fluorescent proteins. While deciding what to use as a reporter for our detectors, we propose the question, which fluorescent proteins are most sensitive? Next year, we anticipate testing several of them and comparing our sensitivity results. Also, we are interested in combining several different sensors into one plasmid to make a single detector that is responsive to multiple heavy metals. in order to tell which promoter was activated. We plan to pair each promoter with a different reporter. For example, what color of fluorescence is produced when our arsenic and lead promoters are activated. In order to increase safety, we also aim to incorporate a cell suicide gene that was created by the 2007 to 2010 University of Hong Kong IGEM project. Fixing the lead promoter may require the isolation of a lead binding protein coding sequence. Development of a biobrick containing a binding protein will require significant additional research. Addressing these issues will be the basis of our project next year. Our work this year would not have been possible without the support of our sponsors. We would like to take a moment to recognize Sandra Bill Hall, Yvonne and Shannon Family, Autodesk, Gaston Day School, Marker Gene Technologies, and New England Biolabs. We will now be happy to answer any questions that you have.
First off, his question was, for our multiplex, we have two different promoters, and one is responsive to multiple heavy metals, and one is responsive to just cadmium. And he's wondering how it's going to be possible for us to incorporate those into one plasmid. And the answer to that is simply, at this point, we've not done enough research into that. We're trying to figure out what would be the most effective way to get that to detect just arsenic. And as you stated, it is also responsive to silver, copper, and cadmium.
The Clemson IGEM team would just take the opportunity to congratulate you and thank you for your work, because through your alumni, Ryan Kane, that Clemson has our first IGEM team and aware of the activity. So we thank you for that.
We would like to thank you also because Ryan Kane was one of the founding members, and without him we would not be here either. Oh. Yes. Every year our school has something called an international festival, and that is where we have vendors set up different booths, and we decided to set up one. And we were expecting high schoolers to actually show up to the event, and we were hoping to be able to explain what a plasmid is, what is IGEM, what is synthetic biology. However, most of the students were 8, 10, 12 years old, and at that point they were not able to actually comprehend what we were saying. So I mean, it would have been fantastic if we could have introduced them to that idea, but because of their age, they've never even taken high school biology, so that's just not an option. Any other questions? Yes, sir.
Could you guys explain just in brief detail about how you quantitated the fluorescence level of your detectors?
Yes, sir. He asked about how we can further quantitate the fluorescence of our detectors, and the answer to that is we this year actually got a new spectrophotometer that has a built-in fluorometer. So at first we took all of our detectors, including the negative controls, and we measured the fluorescence on its own, and we recorded that data. And then afterwards, we also measured the OD 600 reading, and that would give us the growth density, and then we then divided those two numbers. So the fluorescence reading divided by the OD 600 reading would account for the growth density. And in some scenarios, once again, as you stated, with us running into problems and then having solved them, we noticed that all of our detectors were maxing out at 1.016. And so we're like, how is this possible? Come to find out, we needed dilutions, and then we used serial dilutions to find out fluorescence. Yes, sir. No problem.
What was your main motivation to take this project?
Our main motivation is the fact that right now, we are very limited in what we can do, and as Audrey stated earlier, we are completely independent of a hospital or any university. And because of this, we're not able to make our own parts, and we have to look at things that are available in registry. And so at this point, a feasible project for us was simply to tie the promoter with the reporter, and we looked at problems associated with that. And right now, in both the United States and around the world, that is a significant problem, because everyone has water, and lots of times it's polluted.